My name is Ali Mohd. I go to Flowing Wells High School and I studied with Dr. Vanessa DePaz in IBD research under Dr. Paulo Coelho in pediatrics. So the main focus of this lab is to study the causes of inflammation in the small intestine and colon during IBD. And our main question is, what is causing inflammation during IBD? In February, my mentor published a paper that went over the effects of a deficiency in disabled homolog 2 in dendritic cells, or DAB2 for short. DAB2 is a protein that regulates signaling pathways between cells, including pathways that regulate cell growth and inflammatory response. The paper concluded that a deficiency in DAB2 increases stimulated inflammation in the colon. So this is a dendritic cell, or DC for short. Dendritic cells play an important role in immune response, especially in the gut, because they recruit effector immune cells and help develop regulatory T cells, which facilitate tolerance. So earlier this year, the lab published a paper that went over how certain stimuli can decrease DAB2 in dendritic cells. And a decrease in DAB2 in turn makes dendritic cells give off a more inflammatory immune response and reduces tolerogenic effect when sensing bacteria stimuli. And this is what helps contribute to IBD and other chronic bowel diseases. So now we have to find out what is causing DAB2 to drop in dendritic cells. For further testing, we have to collect more dendritic cells. So instead of using the intestinal-like bone marrow-derived dendritic cells used in past experimentation, we decided to try out a new line of dendritic cells called DC2.4. DC2.4 is easier to reproduce and cheaper to use, making it a good alternative to bone marrow-derived dendritic cells. Since they are different cells, however, we have to make sure they exhibit the same properties as the previous cells. So for this experiment, we needed to reproduce a lot of cells because we had a lot of samples to make, and we can reproduce a lot of cells by cell culturing. And during cell culture, I pass the cells from container to container in medium every 2 days to make sure that they don't overconfluate or overpopulate and that they stay healthy. I also check them under a microscope every 2 days to make sure that they're growing right and making sure that none are dying excessively. So after enough cells are reproduced for experimentation, they're split into 6 different groups of 100,000 cells each, and bacterial stimuli is added to them at different time intervals, from 24 hours to 0 hours of exposure. And if the cells do exhibit the properties that we need, then they will show a drop in DAB2 as exposure prolongs. To measure DAB2 levels in our cells, we can use Western blot, which is a protein quantification technique, and RT-PCR. And if the cells do show this drop in DAB2 as bacterial stimulus is prolonged, then we can move on to the next steps. If the cells are viable for experimentation, then we can answer the questions: what proteins cause DAB2 to drop in dendritic cells, and how do autocrine stimuli affect DAB2 in dendritic cells? However, if the cells do not fit our criteria, then we have to ask the questions: why don't they show a drop in DAB2? Are there any uses for these cells? And where can we find cells that do fit our criteria?